Welcome back, everyone. We have a very special guest with us today. We have the wonderful Dr. Tomás Duraj. Thomas, thank you so much for being with us today.
Thank you, Ben, for having me. It's a pleasure.
Well, the prep for this one was lengthy because you've done so much wonderful work, and reading your studies, I learned a bunch and have loads of questions, and no doubt our listeners do as well. The sort of metabolic approach to cancer is very top of mind for our audience. I want to start at the very top, at the sort of the broader level. Could you explain what the somatic theory of cancer is, and then the metabolic theory of cancer is?
All right. Yeah. So maybe if we give a little bit of a historical perspective on these things, I think that is easier for people to kind of situate themselves in what the difference is between like the data that you can obtain in research. Most papers that are published, data papers, experimental papers, in cancer, they can use, cell models, cell culture models, or they can use animal models. And usually what you see is that you obtain a set of data, just graphs with things go up and down within the experimental conditions. And these are then sort of joined or combined into theories, which are a way to try to explain, these sort of data sets. And I typically tend to look more at the data rather than what the authors write. And I think we are a little bit guilty of this ourselves, and every researcher is to a certain degree, to try to make a story out of the data, because to publish your data, you need to make a neat-based, coherent story. Going back in history, I guess the prevailing paradigm right now for what cancer is, more to be more concrete, how cancer originates, is the somatic mutation theory. The somatic mutation theory basically started with Theodore Boveri. He was studying kind of sea urchins, similar to Warburg actually, and he observed that there might be like chromosomal abnormalities in the chromosomes, which are the part of the cell that contains our genome. During cell division, during proliferation, he observed that maybe there is some breaks or some problems with the chromosomes. So he postulated, he wasn't really sure, but he said, well, maybe something with the chromosomes is responsible for cancer, the origin of cancer. And then, of course, with the discovery of the DNA structure by Watson and Crick and Wilkins and Rosalind Franklin, the idea became to be that there might be damage in the genes. The genes that codify our proteins might get ATCG in the structure of the DNA. There might be a break or a change in the nucleotides, and this seemed to lead to dysregulated growth in the cells. This was followed by the one-hit hypothesis, the two-hit hypothesis, the Knudsen hypothesis, which was basically, maybe he was looking at the retinoblastoma gene, and he said maybe you need like two copies of the DNA, the genes that codify for your proteins. Maybe you need like two damages, two loss of function in your alleles to induce cancer. But of course, this didn't always hold. So then the story gets more and more complicated. Then maybe they postulated that you need driver genes, which are the ones that drive the tumor, compared to the passenger genes, which are just mutations in passenger genes that are there, but do not really contribute that much to the dysregulated proliferation. And then Bogustin and Tomasetti basically postulated sort of like a bad luck hypothesis, or the dark genome hypothesis, in the sense that the more and more you sequence the genome, looking for in tumor samples, the more mutations you see. You often see, this is from the Cancer Genome Atlas, you often see like 50 up to 100 different mutations, and these are different between individuals, these are different between different tumors, and these are different between the cells in the tumor itself. So looking at all that, sort of I think the state of the art right now is that cancer is caused by somatic mutations, in oncogenes or tumor suppressor genes. Yeah, these are driver mutations. There is also passenger mutations, but it is not enough to have one driver mutations. In some cases, you need more, and sort of ad hoc, post hoc, I guess. There's this attempt to try to explain cancer, going backwards from the mutations that you observe. I think this has led to some successes in the sense that the idea with this was that we would be able to sequence all the genome, and then we would be able to develop therapies targeted to these specific mutations. There's the Gleevec, the imatinib for CML, which was like a very successful therapy for chronic myeloid leukemia, and the HER2 positive, for example, breast cancer tumors. These can also be targeted with trastuzumab. But other those very specific single gene sort of alterations, unfortunately, in the clinic, all this approach of looking at the genes doesn't really work. You cannot really develop that many therapies based on this. And so I think Dr. Seifried is usually much more eloquent in explaining the failings of the somatic mutation theory. I would, for my part, I would just say that it doesn't seem to work in the clinic. It seems to be, even looking at the data, if you take a step back and you forget about theory, but you look at the data, I think if you don't have a preconceived notion that cancer is a genetic disease, in the sense that it is caused by somatic mutations in oncogenes and tumor suppressor genes, of course, genetics are involved in the malignant transformation of cancer cells. But that is different from what the theory postulates. If you take a step back, you would look at all the heterogeneity and you would say, maybe this is like a more of a consequence of the cancer being formed rather than the cause. And in the hallmarks of cancer, what is cancer? I think maybe we could also start there. Cancer is just uncontrolled proliferation. Weinberg and Hanahan Weinberg in the cancer hallmarks, they try to describe sort of the basic characteristics that cancer has. This was a very highly cited paper in the 2000s, in the year 2000, perhaps one of the most cited in cancer biology. And what they did is basically just describe how cancer behaves. It evades apoptosis, it proliferates constantly, it has proliferation signaling, it doesn't respond to growth signaling, arrest, metastasis, angiogenesis. The cells are immortal. But these are just descriptions that they didn't even mention in the beginning. They didn't even mention mutations. Then they had some revisions in 2011, I think, and then recently 2022. So they kind of keep updating these characteristics that tumors have. And there is now many like core characteristics and enabling characteristics. But that I think that doesn't tell you really what the primary cause is. So I think we would look more and we would be more convinced looking at the data that perhaps cancer, the origin of cancer is mediated by mitochondrial metabolism. I don't know if that makes sense. Maybe we could go a little bit deeper into that.
It does. It does make a lot of sense. I think it's interesting to look at it from the historical context of the discovery of the the double helix. Everything was very focused on genes, right? And I'm in Tripping Over the Truth. There's a lovely chapter on when well-intentioned, brilliantly smart people tried to sequence the genome, and at the end of it, I think it was Steve Jobs's oncologist said, we should just start throwing darts at it because it's too hard to understand, right? Which seems very defeatist, and perhaps we're looking in the wrong place. In the UK, I'm here in the UK. I know you're Spanish, right? But you're in the States at the moment.
Well, I grew up in Spain. I'm originally from the Czech Republic. I studied medicine in Spain and my PhD, and now here in Boston.
Oh, sorry, my apologies. I did Spanish. Okay, right. So you've been around the world, so I don't know what it was like where in the different geographies, but in the UK specifically, where I am, I remember growing up and you hear about someone who gets cancer, and it's considered a sort of it's luck. If you get it, it's luck. There's nothing you can do about it. So what would you say to people who say, oh, but what about the BRCA gene? You know, Angelina Jolie had her breast removed because of it, and you know, surely that plays a massive role. If you've got that, it's basically a death sentence. What would you say to that?
Mm-hmm. Yeah, those are the germline mutations. So this means that these are inherited mutations. What we would say to that, and even the somatic mutations, they can be involved in the malignant transformation process. That would be mainly because they alter mitochondrial function too. So you can inherit, BRCA1, a leaf many, p53 mutation, a Lynch syndrome, all these satellite repair mutations, all these can predispose your cells, and usually it's a pocket of cells. For example, in neurofibromatosis type 1, you would have like the neural cells. These cells could be predisposed to, in our mind, to damage. Well, damage is sort of loaded term. They are predisposed to lose function in the mitochondrial metabolism. Looking more at the bioenergetics, at the sufficiency of oxidative phosphorylation to keep the cell differentiated. And if you inherit the mutation, which it is sort of bad luck in the sense that it is not determined by the lifestyle or anything, just the changes of the genome. These can predispose some pocket of cells in the body to be more prone to have a higher risk of malignant transformation. But this risk is never 100% penetrant. So even the highest penetrance, which means that you would get cancer during the lifetime of the patient, they are typically not 100%. And so we think that means that this has to be mediated by mitochondrial metabolism, in the sense that if for whatever reason you also have conditions in which the pocket of cells that would be most predisposed to develop the malignancy maintains normal mitochondrial function and sufficient oxidative phosphorylation during the lifetime of the patient, then those cells, despite the mutation and even the high risk of developing the tumor, they do not express the phenotype of malignant transformation, because the mitochondria is functioning properly. It doesn't express OXPHOS insufficiency and substrate phosphorylation dependency, and we can define those terms. And as long as it doesn't do that, then the cell will behave as a normal cell. It will not express the hallmarks of the phenotype of a cancer cell.
That's super interesting. So if I hear you correctly, and me trying to sort of make sense of it, it's if you have a BRCA1 or BRCA2 gene, something like this, it's not a death sentence and it's not deterministic that you will get cancer. However, it might lead your mitochondria make your mitochondria more susceptible to dysregulation down the line.
Correct. Yes.
Okay.
And the problem there is that really the factors that would determine whether the cancer phenotype will express or not, I think we have some ideas, but I don't think this has been like properly studied. Mainly because this idea of mitochondrial involvement into the malignant transformation is it's part of the literature, part of the scientific literature, but it is not receiving as much attention as it should in my mind.
I
And I
actually
think
think
if we did these studies, and if we did these, even like in epidemiology, you could look at different factors that might be involved in mitochondrial health, exercise and healthy diet and proper sleep and all these things, and see whether these actually make a difference between the patients that inherit these mutations. And you would have to take tissue samples, you would have to take biopsies of the patients, look at the mitochondria, because it's very difficult to measure mitochondrial function. So you would have to look at the structure or measure indirect markers as oxygen consumption, some of these things, to try to figure out the efficiency of the mitochondrial population, which is very, very hard, even in research. So the long answer is we don't know what to do, even though we know that it is good to keep mitochondrial function. I mean, if somebody does like a 23andMe or well, that company I think is no longer, but
yeah.
some genetic testing, genetic screening, and they see some sort of gene predisposition for cancer. I think, of course, it makes sense to do lifestyle wise as much as possible. And I would also say screening would be very important, in the sense that if there is any incipient type of tumor, there would be more impetus to maybe implement, ketogenic metabolic therapy, or even part of the standard of care. But I think, in prevention terms, it has to be like a personal decision, but maybe like removing the breasts in someone that, very young person that has, BRCA1, BRCA2. Once again, it's a personal decision, but maybe a very aggressive screening with trying to do as much as possible in terms of prevention. I think that would also make sense. But that it's not like in the guidelines, but I think in my mind, it would make sense.
Wow. Okay, interesting. So I guess what I find fascinating about your answer there is that is an example usually touted to support the somatic theory of cancer. The BRCA gene, right? But yet the actual root cause is mitochondrial dysfunction. So it's almost one and the same. You could use it in either argument, right?
Yeah, sometimes we hear that argument. Also, you would hear, or more knowledgeable people in the field, if you argue with a researcher or a scientist, they would also say, one of the most elegant experiments that you can do to induce cancer, or to prove that cancer would be, it would look like it's a genetic disease. It's just basically with like a viral transfection of oncogenes. This is what Weinberg and Prada did, I think, in the 80s. And these are seminal, studies. They were looking at, you have, for example, fibroblasts from embryonic fibroblasts. They are not transformed. That means that they are not cancer cells. So you have like normal cells in a petri dish. And then what they did, for example, is like you can induce. Today you would do it with like a oncogenic virus. You can induce mutations, or you can directly insert a mutated gene. For example, MYC, the MYC gene, c-MYC, which predisposes the cancer cells the normal cells to proliferate, but they are not malignant. So you can have a highly proliferating cell that is not malignant. That means that doesn't form tumors. These are the immortalized cells, and there's like many that people use in research. I think they are still not normal, but we would say that the damage to the mitochondrial apparatus was still insufficient. And then you can also co-transfect them with, for example, KRAS, or the RAS oncogene, RAF oncogene, and this leads to malignant transformation. So, looking at it from what they would look at is of course, you have a normal cell, then you insert a gene, a MYC gene that is always active, then you insert a RAS gene, KRAS or something, and suddenly you have cells that are highly proliferating, and you put them in an animal and they form tumors. So, that just looking at that, you would say, oh, well, I put some genes in, some oncogenes, it starts to proliferate, must be a genetic disease. However, looking at the nuclear transfer experiments, and just simply like the mitochondrial function in this process, where when you are inserting these genes, these oncogenes in the cell, in the normal cells, there is a big drop and loss of function, because these genes are colocalizing in the mitochondrial metabolism, and they are involved in the mitochondrial metabolism. So as long as these genes alter or induce, in my words would be induce a loss of function in the mitochondrial, in OXPHOS, in oxidative phosphorylation, and they induce dependency on substrate level phosphorylation, then you would have the expression of cancer cells. If they don't do that, if you have some fibroblast or something that just simply doesn't happen, you have the oncogene, but you don't induce these changes in oxidative phosphorylation, then you don't have, if they cannot switch to substrate level phosphorylation dependency, which means that, the Warburg effect and mitochondrial substrate level phosphorylation, if they don't make the switch in metabolism, they simply don't behave as a cancer. I think it's very hard in in the chicken and egg kind of situation. In the beginning, I think we could gain some clarity, and it would be great if there was more research interest. I actually have some grants that we'll see what happens, to sort of do more and repeat the nuclear transfer experiments to see whether this is more broadly applicable to all different cell model systems. But I think if there was more questions, if there was more research on this, we could have the answer. If all the research community was trying to figure this out, I think in 5 years we could have the 100% answer either way. There wouldn't have to be a debate or this conversation. But unfortunately, so it's the assumption that cancer is simply this, and I learned that too, in medical school and my PhD. It's very hard to to go against the grain and say, oh well, maybe it's all mediated by mitochondrial function, which seems to be more as a nuance to the thing. But in my mind, just to summarize, changes in mitochondrial function alone, and we can define these changes in mitochondrial function alone are necessary and sufficient to induce malignancy, to induce a tumor cell, whereas somatic mutations are neither necessary nor sufficient. They can be present, they can be involved in the process, and of course, if you have a tumor cell, eventually it will accumulate mutations because of the reactive oxygen species and the oxidative stress that it's under, and the hypoxia, and everything is sort of messed up after a while. So in those, in that microenvironment, of course, it accumulates mutations. Some of these mutations might be helpful for the tumor cell in the sense that the selective pressure in the environment might select those cancer cells that adapt, in quotations, as long as they have fuel, adapt to hypoxia or adapt to invasion or metastasis. So it is involved in the process, but in the beginning, I think they are neither necessary nor sufficient. That's why you see mutations in driver genes and oncogenes and tumor suppressor genes in normal tissues that never became cancers, and you have no mutations in some tumors. Of course, it's a very small portion, but these two things are hard to explain from the SMT perspective, but much easier to explain from the mitochondrial metabolic theory perspective.
We also have time as a variable, right? Of how many years, yeah, right? How many years have been spent on the somatic theory of cancer, and where have we got to? Which is a interesting.
Yeah,
interesting
I think
point.
just to complete, historically, somatic mutation theory or the genetic theory of cancer, it wasn't the only one. I think you mentioned Travis' book. He goes through these different ideas. The viral theory of Peyton Rouse with the Rouse chicken sarcoma virus. He also, for example, he was postulating that maybe it's like viral infections that cause tumor cells to appear. The tissue field organization theory by Sonnenschein and Soto, they are more interested in looking outside the cell, so looking at the sort of the morphogenetic fields, which basically means like the structure, sort of like in embryogenesis or developmental biology, looking at the structure of the tissue to see that when there is loss of structure outside the cell, like a wound, for example, you could have more susceptibility to develop cancer. And the mitochondrial theory, which would be more like the metabolic theory, I guess, because in Warburg's time, the mitochondria wasn't like fully described. This was the grana, which was some pockets in the cell that looked like might be responsible for the energy generation. So Otto Warburg, in the at the beginning of the 20th century, he was looking at the metabolism of normal cells and cancer cells, and he was observing the changes. That's the famous fermentation, or the Warburg effect, later defined. Looking at the changes in metabolism, he got the Nobel Prize in 1931, I think, working in cytochrome C oxidase. So he described sort of part of the electron transport chain where oxygen was consumed. So he wasn't really recognized for his work on cancer. But then one seminal paper in Nature on the origin of cancer cells in 1956. In this cancer, he argues with other researchers in the field whether cancer is directly generated or initiated by changes in metabolism, basically. I think he got the idea. The idea is correct, in my opinion. Some of the nuances were not completely correct, in the sense that he put a lot of emphasis on the Meyerhof quotient, and on the relative changes in between like lactate production, which is the fermentation, which is sort of taking glucose and instead of fully oxidizing it, excreting part of it as lactate, and the oxygen consumption. These things are quite difficult they are all like relative changes. And with his collaborators, with Dean Burke, they have this massive paper where they went through like many tumor samples and normal tissue samples, and they were just trying to say, oh, maybe the basal oxygen consumption, maybe it's a little bit lower, and there's always fermentation. Whatever you do, there is always more lactate production, which is technically true, I think, to this day. But it was a little bit um, confused or a little bit difficult to, because other researchers did the same thing and they said, oh, look, the oxygen consumption is still there, so must be that oxidative phosphorylation, the generation of energy through the mitochondria is still working, and it's fine. So they were kind of arguing back and forth for 30 years, and then poor Warburg died. And then sort of his work was partially forgotten, just because there was this big nascent field of genetics, and not to discredit or not to not put enough emphasis, or like sequencing the human genome is like a miracle of technology and human ingenuity, and I think can and should be very helpful when the technology gets there for these Mendelian diseases, hemochromatosis and anemia falciforme, and some of these single gene problems where it's very difficult to like fix the genes. I think if we could fix the genes in those instances, we could technically fix the disease. I mean, it's just a gene that leads to an aberrant protein. If you catch it early and you could like revert the situation, you would have a normal protein. A normal protein would do a normal function, and you could fix the problem like that. For cancer, if you have 100 genes that are damaged, that have a mutation, what do? Like you can't fix that at the gene level. So Warburg was looking more at the genome. Sorry, I was looking more at the metabolism, and also from an evolutionary perspective. So I typically, maybe in a kind of long-winded story, but typically I like to go back and look at the evolutionary kind of history or the evolutionary biology of the cells. So maybe we could also discuss a little bit that, because I think it explains, and I'm sure you will have many questions. And this is perhaps like the most dry part of this conversation, but I think these many questions can be answered if you take a step back and you look at why a tumor cell would start behaving like a single cell organism.
That was my
Why a
next
tumor
question
cell
anyway.
would.
So
Sorry.
that was my next question anyway. So it's perfect.
Yeah, sure. Going through that, I think the life on Earth, sort of, we were not always, if you look at me and you can see clearly that under the microscope, if you look at it, even without it, I mean, unless you would consider that we are formed by one single cell, we are clearly formed by multiple cells. We are metazoans. We are multicellular organisms, and each of our cells, the dermis or epidermis or different organs, each of our cells sort of live in a society of cells where they are hyper specialized, and they have, there's tissues and there's organs, and they have different functions. But if we go back in the evolutionary history of life on the planet, 4 billion years ago, if we look at the last universal common ancestor, that's the assumption going to the phylogenetic history of life on the planet, is that all life on the planet right now evolved from this last universal common ancestor, which would be like some sort of unicellular organism that then sub-specialized or like evolved into different other different organisms. And this last universal common ancestor was basically single cell bacteria, sort of bacteria, unicellular organism. I think there was this recent paper by Moody et al. In 2024, where they sort of hypothesized that, looking at, there's no like direct evidence of this, but you can look at the other different organisms that would be similar, that still sort of didn't continue evolving that much. And this organism would probably have glycolysis. So the same as Warburg was looking at, there's like very active glycolysis because there was no oxalogs. If we imagine the planet, 4 billion years ago, it would look like a volcanic sort of planet, but there is no trees, there is no, not much life happening. So glycolysis, it had like a ATP synthase on the membrane. Maybe part of the TCA cycle, some of the enzymes of the TCA cycle I also think, reading that paper, I think maybe even mitochondrial substratal phosphorylation, sort of, some of the enzymes involved in that might have been present in this organism, but that's a little bit of speculation. So this organism, basically, the only thing that it could do is divide, as long as there was fuel in whatever environment it found itself in. And I think this puts a little bit more emphasis on the idea that, of course, it had some sort of genome or DNA, because you need to pass it to the next generation of if you are a unicellular organism. But the baseline for life, in my mind, if you make a pyramid of life, on the baseline, you will have metabolism before you have information, before you have genes or information. You have to have energy. You have to have sufficient ATP, sufficient energy to even, before you even try to like divide or pass information to the next generation. So I think that life evolves first as metabolism, as baseline metabolism, that start to form structures that then proceeded to evolve into coherent sort of organisms. So when oxygen came into the atmosphere, 2 billion years ago, by our friends, the cyanobacteria and other photosynthetic organisms, that's when 1.5 billion years ago, the idea is, the endosymbiotic hypothesis, which is that an alpha oxidative alpha bacteria merge with a prokaryotic hypoxic precursor. This is, a little bit concrete terminology, but basically means that the mitochondria, so the precursor of the mitochondria, merged with sort of this hypoxic, fermentative organism, and these started to form the eukaryotes. So eukaryotes are our cells are eukaryotes. This means that we have a nucleus, and we have mitochondria in our cells, which are responsible of generating most of our energy. And then we have organelles and other cellular structures. And it is only after 1.5 billion years ago, when there was oxygen in the atmosphere, and these oxidative alpha proteobacteria were engulfed into this prokaryotic precursor. Only then, when our cells became eukaryotic, we could I guess we could have enough energy to subspecialize to start forming multicellular organisms. So you have, of course, eukaryotic cells that are unicellular, and we can also be multicellular, but you don't have prokaryotic cells, which means you don't have cells that have no mitochondria, forming multicellular organisms. There's some biofilms and stuff, but the only cells that cooperate and that can like subspecialize are eukaryotic cells. So this is like a very long way to say that I think, and looking at sort of this atavistic picture, if there is damage to the mitochondria, which is what is keeping the cell cycle going, keeping the apoptosis in check. Mitochondria are in charge of the calcium gradient and apoptosis, which means cell death. So the mitochondria basically are making energy, and as long as they make enough energy, our cells, eukaryotic cells, behave as eukaryotic cells. If there is loss of function in the mitochondrial metabolism, specifically in the ATP production, the cell has two options. One option is if the ATP-ADP ratio drops, which means that, there's no ATP, there's no energy, they can commit suicide, they can commit apoptosis, which means, this cell is no longer part of this society of cells. It's losing function. Let's kill it, and let's let the other cells around it to maybe divide a little bit and supply this function. Or if it doesn't die, then it could become malignant cell. Malignant cell would be a cell that simply lost function in mitochondrial metabolism enough, not completely, but enough to become dependent in substratal phosphorylation. This is like this ancient old sort of in the history of life on the planet, this old phenotype of metabolism, which is in the absence of oxygen, you can still make energy, generate energy in glycolysis and in mitochondrial substratal phosphorylation. So it is this switch in my mind that sort of determines the malignant transformation.
That makes a lot of sense. I think what you've tied a thread for me that I haven't heard before, which is I've heard lots of people say most diseases are metabolic in nature, and I'm inclined to sort of agree. But you know far more about it than I do. But I think you've just tied the thread for me. So if I hear you right, from first principles, us as an organism require energy before anything else, right?
Correct. Yes.
Okay. And when that metabolism, our energy goes awry, problems happen downstream of that. In cancer is one of those.
Yeah, I think across the different spectrum of diseases, I mean, not all diseases are, are caused by lack of energy, of course, like an infection or something,
Mm.
or um, yeah, not all disease, even like maybe the monogenetic Mendelian diseases, where you simply have like a loss of function in some other protein that might be involved in something completely else. Uh, those are not, not
Yeah.
involved in energy, but for most like the metabolism based diseases, for example, like neuroprogressive diseases, neurodegenerative diseases like Alzheimer's. In that case, basically our neurons, if you take a look at where you get cancer, it's usually in the epithelial cells or it's in the stromal cells, the support type cells, like fibroblasts or mesenchymal cells or stem cells. So those are the cells that when they lose function in oxidative phosphorylation, they can make the switch to substrate level phosphorylation dependency and they can become malignant. Are terminally differentiated neurons or maybe myocytes or even muscle cells. You don't typically see cancer cells there, and that's different from like a glioblastoma, for example. Like a brain cancer, people imagine, oh, must be my neurons are forming a cancer. It's not neurons, it's glial cells or other supportive cells. So usually you get cancer in epithelial cells and supportive cells. In our neurons, if we have loss of oxidative phosphorylation, typically the neuron dies. So in Parkinson's, you would have in the substantia nigra, you would have neurons that are dying. In Alzheimer's, you have neurons that are dying, and this causes, of course, by loss of mitochondrial function, you have loss of function of the whole organ. Of course, insulin resistance, diabetes, these are also involved. That's more like an overflow of energy where you get problems with sort of the buffering of energy. And you hormonally, you also get problems with too much insulin that signaling too much, but then the cell cannot adapt and you get problems from that. So all these diseases, yes, these are more mediated by mitochondrial metabolism. And I think once again, maybe not to miss the forest for the trees, but in many of the standard treatment options for all these that I mentioned, including cancer, this is like completely out of the picture. There's like no consideration for mitochondrial metabolism. For one reason is because it's quite difficult, as I said, to fix or to even measure these things properly. So usually what you get is like you remain at the most superficial level. You say, oh, I have diabetes, my sugar is high, so I use insulin to push the sugar down, and that must be the solution. Instead of looking at why is insulin insufficient to keep your sugar in check. So yeah, I think there's still a lot of work. If we wanted to treat these diseases properly, looking at their origin, I think there's still a lot of work that needs to be done to postulate. Many of them can be solved easily, with lifestyle changes and diet changes. So maybe there's even no need to to do much research. But for the ones that do not respond to this, or for the people that choose not to do some of these changes, then there could be therapies that could help. But
Yeah.
I think this is like a long research arc that still needs to happen.
So I'd love to get into the sort of the bioenergetics of a cancer cell and how it uses energy, what kind of energy it uses. But before we get onto that, and it's kind of an adjacent question, the Warburg effect, right? That's well appreciated in all circles of cancer, to my understanding. When it comes to the cancer diagnosis, right? My understanding is a PET scan is a direct confirmation of the Warburg effect in uh, standard of care currently. But yet, when it
Yeah,
comes to treatment,
yeah,
sorry, but when it comes to treatment of cancer, that's completely not regarded at all. Why do you think that is?
yeah. You would think so. Looking at the PET scans, 18 fluorodeoxyglucose PET scan, that the Warburg effect would be appreciated. But it's really not, in the sense that there's sort of like a counter trend in the research field. I personally believe it's a very important marker of the metabolism. But you could argue that the FDG PET scan, basically what it only indicates is that there is higher uptake of glucose in the tissue. It doesn't really answer the question whether this glucose is consumed by, it's going into the Warburg effect, which is like a different metabolism of that glucose, or whether it's also partially oxidized. Of course, it's partially oxidized. So, I dislike sort of the term Warburg effect a little bit. And in our research, when I came to the lab and with Dr. Seifried, we actually have a new paper that is coming out that he's very excited about, trying to like summarize many of the misconceptions and the questionable or incorrect assumptions that people have made in the field of metabolism. So look forward to that. But
Yeah,
we are trying
sure.
to redefine the Warburg effect. This was coined by Ephraim Racker in 1972. And basically, I think the problem with the Warburg effect is that it confuses. There's like three variables that make it difficult to know what's going on. You have the initiating metabolite, so you have glucose. Glucose goes into glycolysis, and it's transformed in different enzymatic steps. It arrives at pyruvate, and pyruvate can either go into the mitochondria to undergo oxidative phosphorylation, or it can, through lactate dehydrogenase type A, it can go into lactate. And the Warburg effect would be basically the continuing production of lactate. So it's, instead of the glucose being oxidized, it is fermented. Fermentation is just basically this step to lactic acid fermentation. So it goes into lactic acid when it leaves the cell and bounds to hydrogen, even in the presence of oxygen. So in the presence of oxygen, you would expect complete oxidation of glucose, but it's like a double negative. Instead of the expected oxidative phosphorylation of glucose, you see lactic acid fermentation and you're also looking at the initiating. So you're looking at the initiating metabolite, which is glucose. So it's like a little confused. I think it doesn't tell you anything about ATP energy. Like the Warburg effect doesn't produce energy. The Warburg effect, technically, it's more involving the NAD plus regeneration at the end of glycolysis. So NAD plus is reducing equivalent. And it's consumed during glycolysis, and to replenish it, pyruvate to lactate can replenish NAD plus. So I think I prefer the term substrate level phosphorylation. So substrate level phosphorylation is simply the description at the most basic enzymatic level, where you have a substrate. It is transformed by an enzyme into a product, and during this step of transformation through the enzyme, ATP plus phosphate give you ATP, and that's all you need to know of during the Warburg effect during glycolysis. The Embden-Meyerhoff-Parnas pathway has certain enzymatic steps that consume ATP, hexokinase and phosphocryptokinase, and you have other steps that you do this twice. So the net benefit is two net ATP molecules, to phosphoglycerate kinase and pyruvate kinase. That's the net result in terms of energy. So substrate level phosphorylation flux in glycolysis would be simply the amount of energy that is produced, during a high flux through glycolysis gives you a lot of ATP, because it's a very fast process. So instead of Warburg effect, I do prefer talking about increased substrate level phosphorylation flux, or substrate phosphorylation dependency, which may or may not be accompanied by the Warburg effect or the Crabtree effect. In cell culture, when you have cells growing in cell culture, in outside the body, most of the cells, even normal cells, not only cancer cells but even normal cells, do the Crabtree effect, which is like a transient Warburg effect, in the sense that they make lactate even if they have enough oxygen, partially because of the oxygen pressure, because it's not the same growing in like a liquid outside the body, partially because of the high glucose concentrations and the competition for ADP and phosphate. So that's a separate issue, but basically the Warburg effect, I don't think it's a good terminology, and we are trying to move towards the term oxphos insufficiency. This means that we don't say damage. We shouldn't say damage. I know reading comments and seeing what people sort of take away from these conversations, and I know they can get a little bit technical. We shouldn't say damage or dysfunction, because that's like doesn't mean anything. Like where is the damage? What's going on? So I think it's better to say oxidative phosphorylation insufficiency or sufficiency. So our normal cells have oxidative phosphorylation insufficiency. This means that most of the energy that they derive comes from oxidative phosphorylation inside the mitochondria through the electrochemical gradient and the electron transport chain creates this proton gradient, and then complex 5 makes a lot of ATP very efficiently in oxidative phosphorylation.
Okay.
Now, if there is damage to this process, and this one of the sort of advantages from a conceptual level of the somatic mutation theory is like it's very clear. You have just a change in the nucleotides, ATCG, or like easy. If you say, oh, there is loss of function in oxidation, then you have to look at all the steps that are involved that begins is that pyruvate enters as acid, and this creates the reducing equivalence and ADH to the electron transport chain. So there is like many steps that both in the structure of the mitochondria, which is the efficiency of mitochondrial metabolism, the ATP production is very dependent in these cristae. So if you have loss of cristae, which are these folding inside the mitochondria, if this is lost, if the structure is lost through crystolysis, if any of the enzymes don't function properly at 100% efficiency, you are losing the amount of ATP that you get. It's mitochondrial fission and fusion dynamics. Our mitochondria are not static. We have 100 up to 1,000 mitochondria in each of our cells, most of our cells, and these are constantly dividing and splitting and fusing together and moving around across the microtubules. So if you lose the number of mitochondria, even though they might look great, if you lose the number, you might also lose the total ATP output of oxidative phosphorylation. So if any of those things start to happen, your cell can either die, or the cell, if it's a cell that can undergo the switch to malignancy, an epithelial cell or a stromal cell, it can be more and more substrate phosphorylation dependent, which means that as oxidative phosphorylation is going down, it has to get the energy from somewhere. Otherwise, it will die. So in order to get energy from somewhere, it upregulates the flux through the substrate phosphorylation level pathways. Is predominantly, there's only two. One of them is in glycolysis, is phosphoglycerate kinase and pyruvate kinase steps, which give you ATP. And one of them is inside the mitochondria, which is another problem because we see ATP coming from the mitochondria, but we don't know whether it's coming from OXPHOS or mitochondrial substrate level phosphorylation, which is at the succinate-CoA ligase or succinate-CoA synthetase step. There's another substrate level phosphorylation step in the TCA cycle, in the Krebs cycle, which gives you a little bit of energy too, predominantly from glucose. So, predominantly from glutamine. So glucose gives you a lot of ATP through substrate level phosphorylation in glycolysis, and glutamine and other amino acids, and basically anything that enters into the TCA cycle can give you also a little bit of energy through mitochondrial substrate level phosphorylation. But the amount of metabolite that you need to get enough energy has to be a lot. It can be a little bit of glucose and a little bit of glutamine. It gives you a little bit of substrate level phosphorylation ATP, but that would not be enough to compensate for a massive loss of function in OXPHOS. So there is this chronic shift to substrate level phosphorylation, a reduction in the efficiency of OXPHOS. And as I said, this can be caused by many different factors. So, the oncogenic paradox, this is Albert St. Georgi. This is a very famous researcher. He was sort of puzzled. How is it possible that if cancer is a genetic disease, how is it possible that like different chronic insults that have nothing to do with genetics, how can they form cancer? Like hypoxia or chronic inflammation or some viral infection or some parasitic infection, different things, which in our mind make sense because if they chronically damage the function of oxidative phosphorylation, then it gives chance to this metabolic shift happening. Once again, as I mentioned before, if there is loss of function in oxidative phosphorylation, the only way to respond is either to die or to adapt with a different fuel source, with a different energy source, which would be substrate phosphorylation, which can glucose and glutamine are the primary metabolites, but there might be others, uridine or other amino acids can also contribute a little bit, but they are not very highly present in our bodies, in the tumor microenvironment. And this shift happens to substrate phosphorylation. And if you go back, as I said, to these 4 billion years ago, and we look at this LUCA precursor, that's the type of metabolism that many cells, like prokaryotic cells, are doing. So it's simply expressing this phenotype of uncontrolled proliferation, because when you are a single cell organism, you care only about yourself. You don't care about the other cells. You are competing with other cells. You are not cooperating with other cells.
Yes.
So I think that's sort of how we would look at the metabolism right now, is that different fuels can feed into substratal phosphorylation flux, and cancer cells are dependent on this. So if you try to force a cancer cell to use only oxidative phosphorylation, they typically die or they normalize their behavior. And of course, a normal cell is happy to do mainly oxidative phosphorylation. So that's sort of the difference in metabolism. And I think targeting the primary difference between normal cells and cancer cells in terms of their metabolism, which is the substrate phosphorylation dependency. Cancer cells are dependent mainly on these two fuels because those are the most abundant. That would be like the primary therapeutic intervention that we would consider before doing anything else. Of course, if we target substrate phosphorylation, which is mainly glycolysis and glutaminolysis, glucose and glutamine utilization by the tumor cell, and then you end up having like an indolent tumor that somehow has enough oxidative phosphorylation, so the damage in OXPHOS is sufficient to induce cancer, but not too steep, and you have cancer cells that are using a little bit of fatty acids or a little bit of ketone bodies or a little bit of some of the other amino acids or something, they wouldn't grow, because if you have no glucose, you cannot divide. If you have no glutamine, you cannot divide. But if they would still be there, then we could go into and try to figure out, how to get rid of these. You probably wouldn't even have to target the metabolism anymore. You could probably, a little bit of low dose chemotherapy, a little bit of like any stress to those cells should be able to eliminate them, maybe some immunotherapy. But right now we are sort of doing the complete opposite. We are starting from the most aggressive sort of DNA, alkylating agents or some DNA damage agents that are trying to maximize the DNA or the DNA repair mechanisms that are trying to like kill any proliferating tumor cell, or some targeted therapy that is very effective in killing specific mutations bearing tumor cells, but it doesn't eliminate all of the tumor cells, so you eventually have recurrence. Instead of that, we should be priming the metabolism of the patient with cancer to make it much more likely that they would respond well for all the other therapies.
Well, on that, my mum, I think we mentioned before, got diagnosed with stage 4 colorectal cancer, and she went into a deep state of ketosis, or keeping her GKI around 1, 2.5 months before she started chemotherapy, and actually found out there's a clinical trial going on in Italy where they have two groups, and actually was in colorectal cancer, where group 1 didn't do any interventions with anything ketogenic therapy, and group 2 started it 1 day before. So if I'm hearing you correctly, actually, the earlier you can prime your body, the better.
Yeah, I think one of the beautiful things about, and the CPT always mentions this, one of the beautiful things about sort of this conceptual framework is that sort of metabolic therapy, or just considering metabolism in your timeline, gives you the chance to use it both as a prevention tool. Of course, for prevention, you maybe don't have to be as strict with it, and you don't maybe have to bring in some other repurposed drugs and some other pharmacology or systemic treatments that are like more aggressive or intensive. And then for treatment, you can sort of continue your prevention strategies into the treatment, but of course you have to be a little bit more strict and focus really, in my mind, to measure a little bit better than we have done in the past in previous clinical trials. I mean, I wasn't involved in these, and maybe the technology also wasn't there, but for a trial that is changing the diet, and for the only diet on the planet, basically ketogenic diet, that has a clear biomarker of you are in ketosis or you are not in ketosis, depending on the levels of beta hydroxybutyrate and acetoacetate in the blood. We have done I have to say a very poor job in like measuring in these trials whether actually people are biologically compliant with the diet. I looked in this BMC paper that we can talk about. I looked like at most of the trials that have been done in GBM, in glioblastoma in brain cancer, one of the most aggressive brain cancers. And unfortunately, it's very hard to decipher or discern whether some of the, quote unquote failures, like when some patients didn't respond as we would expect to the ketogenic diets in terms of therapeutic benefit, was it A, because, in most of those trials, these were like very short term. So they did it, for like, one of them actually did it for like 3 days during radiation therapy. It's not even time to like start being ketosis. And, but mainly, a month, maybe 2-3 months, or because despite best intentions, when you are trying to randomize these things, which, we always get hit like, oh, you have to do a randomized clinical trial. That's going to be hard for like a diet, especially like if you want to blind it. And even if you don't want to blind it, so of course, like hiding from the patient that is eating a ketogenic diet, it's hard. But even when you just randomly, you have patients that go into your office and they are dealing with a cancer diagnosis, and you are just randomly offering them, oh, do you want to be a part of the ketogenic diet trial? It's very hard to recruit patients. And once you recruit them, if you just assign them randomly, oh, you go to the control diet, you go to the ketogenic diet, that's not how you can. This is extremely dependent on self-selected patients that are doing this because they want to. You cannot make these changes if you don't want to make them. Even if you have a dietitian screaming at you 24 hours a day, 7 days a week, you just can't. It's just like every single food choice that you make during the day has to be like a endogenous internal decision to eat this but not that. Don't have like non-compliance events. You cannot just have a little bit of cheesecake just because it's some special event, something. I know it's like very strict. Once again, it's compounded by the fact that you are dealing with a serious disease, which of course in the whole psychology aspect also very important. So I think it's very important to work with a knowledgeable, qualified dietitian that can help with all these things. But it's unfortunately it rests on the shoulders of the patients themselves or their family members, but especially the patient. And that's like a very big divergence from how medicine is traditionally done. You just go to an oncologist and they prescribe to you this regimen of chemotherapy and they do the radiation. You sit down, you do the radiation, you do the chemotherapy. It's like, it's extremely, and I sympathize and empathize with, like, it's very difficult situation. So there is a lot of braveness to undergo all this. So that's not a problem. But the problem is it's most of it is externally administered to the patient that's just passively receiving these treatments. And for ketogenic metabolic therapy to work, unfortunately, the only way to do it right now is with making these dietary changes, which need to induce a change, an objective biological change in the metabolism of the patient. If those changes do not happen, even if you are trying to follow a ketogenic diet, if these changes of try to mimic a little bit the metabolism of fasting, if that doesn't happen, despite saying like, well, I'm following a little bit of a ketogenic diet, but I'm not measuring anything, then just might not, most likely it will not work. So that sort of complicates a little bit the practical implementation of all of this. But I think eventually we will figure out. So one of my pet peeves is that I always try to understand the other side. So I understand that many of the criticisms that are levied against it, but I think that the way forward is to have patients self-select. That means that patients themselves decide to be part of a clinical trial, a pilot study, case reports, case series. And then these patients, which are highly motivated and they are following as best as possible, will have to do a better job in measuring better. So anyone listening, if you are also measuring your glucose, your ketones, all this information is very important for us. So anyone that is having a very good outcome, we're happy to hear. And we are, of course, always trying to publish, exceptional case reports on somebody that is doing great. so measuring as much as possible to figure out, all right, there is a consistent level of glucose in the glucose levels in the blood and ketone levels in the blood that are most associated with the best outcomes, depending on the disease, depending on the cancer subtype. I think, of course, as low as possible is best, but we don't know. Maybe if you go overboard and you only fast, you start 30-day fasting and you lose a lot of muscle mass, maybe indirectly that is associated with worse outcomes instead of better outcomes. So there, I'm sure there's like a threshold of glucose levels, ketone levels, which is what we can measure in the glucose ketone index as an indicator that you are sort of doing the diet properly. And then with all the standard treatments and additional treatments that can be uh, incorporated into that, which would be very useful for, I think, most physicians just working in standard oncology and standard practice. They are like, very, give me the guidelines. They are happy to do many things, but they just want to see it written in the guideline. So for them, if we knew exactly, yes, there is 50% increase in survival or something, with like these levels, then all the pharmaceutical industry, which also would be very happy to capitalize on this, I think, we're like, okay, let's figure out interventions and drugs and maybe easier ways to eat the diet to maintain those ranges, because now we know that in these pilot trials, now we know that these work better than just eating cookies and drinking Ensure and
Yeah.
eating as much as possible. So I think that's sort of the for diet part, that's sort of the hurdle and the limitation that we are trying to fight right now. It's just
Yeah.
trying to bring it into a more biology-based, objective measurement standpoint.
But that's why it's so key for conversations like this, I hope, where, like, for example, my mum, when she got the buy-in that she really understood how this is actually going to help her and how a cancer cell actually, switched and uses fermentation and everything we've spoken about, right? The compliance when you have a why goes sky high, right? People are way more likely to follow through with it and understand the parameters of what they can actually do with it. And I think the other important aspect it adds is it adds a psychological element of control. It's become something very external. I have no control over. This is an optimization problem now. If I can keep my GKI at 1, right, I think is the level, the odds are more in my favor than if I weren't to do that, right? Um, I'd love to understand. So I think our listeners are pretty familiar with GKI of 1 and understand uh, the need to be in ketosis, right? And the glutamine piece, I'd love to drill into. Um, Don is apparently the best glutamine blocker, but it's very inaccessible. What do you think of drugs like N-benzodal and fenbenzodal specifically for their application in blocking glutamine? And then I'd love to explore if they have any other pathways associated with better cancer outcomes as well.
Yeah, so I think here we could step back a little bit and sort of conceptualize, as I did in the paper, which I encourage people to read. But I know it's uses sort of the technical uh, wording and the terminology that we use in the field, because it was written more for medical professionals and other researchers trying to kind of establish how future clinical trials in this whole field could be done. But there I sort of distribute all this into dietary ketogenic metabolic therapy and pharmacological ketogenic metabolic therapy. I often get little irritated, I guess, and we do it too. Like when people say metabolic therapy or something like that, that doesn't mean anything. It's even worse because there is like other sort of snake oil people that use these terms before, and they do like some weird stuff. So we have to specify that we are talking about ketogenic metabolic therapy first and foremost. And then when I say that, most oncologists or most physicians would just imagine, oh, it must be that ketogenic diet that I've heard about. But that's too difficult. I don't want to do that. So instead of that, we also want to differentiate between dietary ketogenic metabolic therapy and pharmacological or systemic ketogenic metabolic therapy, in the sense that the dietary ketogenic metabolic therapy, one of the tools that you can use to induce this, basically what you are trying to do with dietary ketogenic metabolic therapy is induce a whole body systemic shift in the metabolism, in the normal physiology, within the normal physiological range of metabolism, to push it a little bit more to behave like during fasting. So you are trying to mimic fasting or recapitulate the metabolism of fasting. And there's different tools that you can use for that. Of course, the most long-term sustainable tool is a ketogenic diet, but you also have caloric restriction. You have fasting. Fasting itself is, of course, the most efficient way to mimic fasting. Long-term fasting, short-term fasting, intermittent fasting, fasting mimicking diets, even exercise. I know it says dietary ketogenic metabolic therapy, but even exercise technically for a period of time on the level of on the molecular level of the cells, normal cells and cancer cells, it's mimicking fasting because like all the glucose is being consumed, even the glutamine can be slightly consumed. And on the signaling level, you are basically mimicking fasting also with exercise. That's why you can have, you can have ketosis induced, ketogenesis induced by exercise, by very heavy exercise. You can start having ketones in your body because the body thinks that you are running out of energy and starts making ketones. So ketogenic diet is just part of this set of tools that you can use to mimic this metabolism of fasting. And this is a very clear objective criteria, which can be, like a proxy marker. A proxy meaning like an indirect marker for this is the glucose ketone index. As you mentioned, your mom measuring the glucose and ketones, trying to aim for GKI of 1 or under 2. These are sort of arbitrary numbers. I mean, Dr. Seifried and Purnam Mukherjee and the team here at Boston College, they did some studies on this, and there's a clear correlation with like the glucose levels and the ketone levels with our models of tumors growing in the animals. But we don't have like a large study that would try to associate, as I said, that's one of the problems, try to associate the glucose ketone index numbers with outcomes across different tumors or even a specific tumor. But from the mechanism and the biology, it makes absolutely sense that as your glucose, basically your glycemia, your level of glucose should be as low as physiologically possible, which doesn't mean zero. It will never be zero. That's another of the sort of criticisms or complaints that you hear any physician or oncologist. I want to lower my glucose. Oh, you can never lower your glucose. You can lower your glucose, of course. Your baseline level of glucose can be extremely stable using a ketogenic diet or during fasting, and your liver and your kidneys through gluconeogenesis will just produce a little bit, 3 millimolar, maybe 3.5, 4 millimolar. They will keep it in a normal range, but your insulin is down and your glucose can be very low, and you compensate for the lost energy from glucose, which all of your cells, your normal cells, which hopefully it's, even if you are dealing with cancer, your normal cells are the majority of cells. 99.9% of your cells are still normal cells, and they are competing for this glucose. So they are using this glucose much more efficiently. So your glucose lower than baseline, your ketones go up, and you can measure this in this proxy marker of glucose ketone index. Now, ideally, I say it's a proxy marker, and I like the concept a lot, but you always, when you say GKI, you always have to think in your head, you are talking about a chronic sustained GKI. So chronic sustained should always be before the term glucose ketone index, or GKI, because otherwise, if you just drink ketones for 20 minutes eating a high carbohydrate diet, you could get a GKI under 2 or under 1, but that is not at the biological level. That is not mimicking fasting. That is, well, maybe slightly. The ketone bodies have signaling properties, but that is not the same as endogenously all your metabolism is sort of shifting into this mimicking fasting and more inhospitable environment for the tumor. You are stealing nutrients from the tumor. You are stealing signaling properties. Insulin is down. Everything is mTOR is down. AMPK is up. PGC alpha, 1 alpha. Fatty acid oxidation is going up. All these things, you are sort of forcing your normal cells to be very efficient with oxidative phosphorylation, and you are reducing the substratal phosphorylation fuels for the cancer cell. So if that is not happening, then you are following a ketogenic diet, but maybe a little caloric excess. So maybe insulin is still slightly elevated, glucose is still slightly elevated, despite the ketosis. I think for cancer, not for other diseases, for diabetes, this is great. I mean, if you reduce carbohydrates, you can basically put diabetes into remission. And I know people get angry about that, but it's in a remission as long as you follow the diet, basically. So it's just the terminology. But for cancer, you might need to be a little bit more strict with this, and don't not obsessive. I also see people take it a little too far, but strict, depending also on the tumor subtype and what you are trying to achieve, to really measure and see that the GKI is in the correct ranges, sustained chronically over time. Yeah, there will be
Okay.
variations, and it will go up and down. So that's for
So
the diet. Yes.
so that's the GKI piece, and I think that's super clear, and you made some super important points as well. When it comes to the glutamine blocking piece,
I
when
was
it comes,
getting there.
oh, sorry, my bad, my bad.
No. Good. So the diet is basically, the dietary ketogenic metabolic therapy is basically a way to prime the body to make your healthy cells more healthy, in quotations, to be more efficient with their oxidative phosphorylation, and to reduce the baseline substrate phosphorylation fuels, which is mainly glucose in that case, with the diet, and all the signaling milieu, insulin signaling and growth signaling and pro-growth signaling is going down. So you basically prime your body to be able to withstand targeting of glycolysis and glutamine lysis using drugs. So in the paper, I sort of separate the dietary part, which is like sort of your baseline, your sine qua non condition that you need to be able to target metabolism, because you can target metabolism without this ketogenic metabolic therapy dietary baseline. Or one day, if we figure out different drugs that could mimic that, I think it's going to be difficult, because the diet changes like thousands of gene expression pathways in the body that with drugs will be complicated to mimic. But you need that to be able to withstand better the targeting of metabolism using drugs. You can use a glutamine inhibitor, and this is how they tested it in the past. Like you mentioned, it's 6-deazoxyphosphonoleucine, DON.
Wow, a fascinating man to listen to. And if you are just as interested as we are in hearing Dr. Tomás Duraj talk further about metabolic ketogenic therapies, please tune into part 2, which will be released on 18th of April 2025. However, if you are listening to this in the future, all you need to do is scroll up and on the list of episodes, click on the next one for part 2. And we'll look forward to seeing you there. Thanks for listening.